in this project, we aim to solve actually two of these really hard challenges, such as the poor migration into tumors, and then secondly also this profound suppression of CAR T cells in the tumor in its microenvironment. By adding this CCR8, we could show in vivo that we have a targeted migration towards its ligand, first of all, this CCL1. And by engineering our CAR T cells with these extra receptors, we could show that we have homing of our CAR T cells in the tumor-draining lymph nodes, and specifically in the tumor-draining lymph nodes. And secondly, also, we could show that we have higher infiltration of our CAR T cells that carry this CCR8, this chemokine receptor, in the tumor itself. And the ligand, CCL1, was discovered by a former PhD and then postdoc here in this group, Bruno Kadia. And what he essentially did, he analyzed the mRNA and the protein expression of CCL1 in different organs from mice. And what was really interesting to see is that CCL1 is really low expressed in organs, but in a tumor microenvironment it's really high expressed. And this is the key insight that we now aim to also to transfer to human and also to more cancer samples, is to really look at this tumor microenvironment and see, okay, the chemokine ligand is only produced by the microenvironment and not by the tissue itself. So it's really the microenvironment that also attracts our CAR T cells. That was the first big thing that we discovered, and that was also highly appreciated, I think, in the ITOC, in the presentation. And then the other thing, which is actually now well recognized, is this dominant negative receptor. We talked about it in ITOC, and we had also a nice discussion that when the CAR T cells are in the tumor microenvironment, they often get suppressed. And then you have this impaired capacity, impaired lytic capacity of these CAR T cells. And by adding this dominant negative receptor, we really can diminish this negative effect of TGF-beta by just absorbing it like a sponge onto CAR T cells. But by adding this dominant negative receptor, we don't have any downstream signaling, so we don't have any of this normally negative influence on CAR T cells. We only have this molecule now on the cell surface, but we don't see any of the SMAD2, 3, 4 signaling. And this is great because you have now these CAR T cells in the tumor that really can produce these lytic enzymes and kill the tumor cells effectively. And that's what could be shown also in numerous mouse models now, for example, by looking at the pancreatic model, namely BXPC3, or also breast cancer models and colorectal cancer. We could also establish, or we could always see a quite nice effect of these CAR T cells that carried this dominant negative receptor. And I mean, the general conclusion of this whole project, and also what we could see in ITOC, was that there was really a great interest in how to transform or transfer this technology from these preclinical studies that we now do in our laboratory and that were initiated by Bruno Kadia, to now transitioning these findings and also these incredible results into somewhat of a clinical relevance. And also, of course, the aim then is to go into clinics, because I mean that's what we finally aim to do, to really overcome these challenges that we face now in CAR T cells and solid tumor therapy. And this, I think, was the great conclusion that everybody was then asking questions: how is this clinically relevant? And we could answer, and I could say, yes, this is clinically relevant, because these CAR T cells might do the job and do better infiltration, and then also have a greater effect in the tumor microenvironment. So, to speak, ameliorate the efficacy of CAR T cells in solid tumor therapy.